we've been considering the regulation of the lactose operon in bacteria, and we've considered the model by which repressor protein shuts the operon off unless lactose is bound, which in our previous discussion involved the loss of affinity for operator DNA and the falling off of repressor when lactose is bound, thereby turning on transcription of the operon so as to produce the gene products required for the breakdown and the utilization of the sugar lactose as a food source. Now, in our discussion so far, there was one major oversimplification, and that concerns the structure of the repressor protein. The repressor protein actually forms tetramers, homotetramers, and it first forms dimers, homodimers, meaning two of the same repressor proteins associate to form a dimer, and then those dimers can come together and form tetramers, are brought together to form tetramers. And in the tetramer, only two of the monomer repressor proteins have DNA binding sites that are capable of binding operator DNA at any one time. So this is the structure of the monomer of the Lac repressor. And you can see that there is a tetramerization domain that is involved in bringing dimers together. And then, ligand binding domains, which also serve to dimerize the monomers into dimers, which then are tetramerized into tetramers. And then here is a hinge and the DNA binding domain of the repressor. So the repressors actually in vivo in the cell exist as tetramers and bind the operator DNA by virtue of two of the monomers of the tetramer recognizing and bind to operator DNA. And the repressor actually in its DNA binding state has non-specific affinity for DNA. So it doesn't bind to the operator specifically until it encounters operator DNA. And then a change in the conformation of the protein occurs such that it changes its conformation so as to grasp the operator DNA with high affinity at the particular nucleotide sequence that it recognizes at the operator. But until it encounters operator DNA, it actually can slide along chromosomal DNA very rapidly. And that way it's not constantly coming on and off DNA looking for operators. Rather, it can slide along DNA and already be associated with DNA until it finds operator and then can bind it very tightly as it changes its conformation to a high affinity conformation. And that allows the repressor then to find operator DNA with high efficiency as opposed to trying to bind DNA here. Oh, that's not the operator. I'll come over here and try that, try this, try this. That would take far too long. Rather, the repressor can slide along DNA until it finds operator for which it is specific. And the change in conformation of repressor when it finds operator DNA is shown in the following morph, which is derived from crystal structures of protein. This morph was produced by Eric Martz, a retired professor at the University of Massachusetts, and is available on Proteopedia, a wiki site for protein structure. So what we're going to observe is the protein changing from its non-specific DNA binding form, where it can slide along DNA looking, if you will, looking for operator DNA. And then once it encounters operator DNA, there's a conformational change in the protein that allows it to clasp the operator with very high affinity and remain bound there unless lactose binds to an allosteric binding site and causes the repressor to fall off operator DNA. So let's look at this conformational change right now, this morph. And what we will see is that what we have here is a dimer. Here's one monomer and here's the other monomer. We're not showing the tetrameric form, nor are we showing the other domains of the protein. We're only focusing in on the DNA binding region of the protein and the hinge regions of the protein. And what you'll observe is that the hinge regions change from a disordered form in the nonspecific binding conformation of the protein to an alpha helical form in the specific DNA binding form of the protein. And accompanying that is a bend in the DNA. The protein actually bends operator DNA. So let's watch this. Here's nonspecific and specific. Nonspecific, specific operator binding specific operator DNA binding, non-specific DNA binding, specific operator DNA binding, non-specific. And you can see the hinge regions changing conformation from an unordered loose form to structured alpha helical form, which actually sits in the minor groove of the operator DNA, and the operator DNA is seen to be bent by the protein. So again, the protein changes conformation when it encounters operator DNA and clasps that DNA quite tightly, accounting for repression of the operon. Now, let's go into the data of Jacob and Monod, two French geneticists who in the early 60s discovered the mechanism of the regulation of the lac operon in some of the most elegant experiments in all of genetics. And I'd like to review a little bit of the techniques they used before we look at their data that they achieved. And their study of the regulation of lactose operon and the deciphering of it depended upon their ability in bacteria to create what we would call marrow diploids. So normally we have the bacterial chromosome, the large bacterial chromosome of approximately 4 million base pairs, 4 times 10 to the 6 base pairs. And there are plasmids that are independently replicating small circular molecules that can replicate inside bacterial cells. And these plasmids can be made to carry portions of the host chromosome. So if we were to blow a plasmid that they used up, you can actually see that in a plasmid, you could create a portion of the plasmid that carried chromosomal DNA from another chromosome, another bacterial chromosome. So this region here represents a gene that would normally be present, a gene or operon that would normally be present in the host chromosome, but is also now present in the plasmid as indicated. And when we have that situation, when we look at this operon, and then there's an operon, the corresponding same operon in the host chromosome, we call this situation a marrow diploid. So we know bacterial cells are haploid, but in this case we've made them, through genetic techniques, we've made them partially diploid. These are called marrow diploids, and they're diploid for whatever part of the chromosome is now carried on plasmid DNA. Now, it's not an exchange. We haven't removed the genetic material from this host chromosome and put it in a plasmid. Rather, this has picked up that region of chromosome from another bacterium, and we can propagate this plasmid bearing a portion of the bacterial chromosome in another host cell, such that we now have a partial diploid for the genes that are represented here in the boxes. And these marrow diploids then allow you to create a situation in which you can have one lac operon. You could say have lac I repressor gene here, wild type. We can have then the operator DNA, or the promoter, and the operator DNA. And then we can have the 3 genes of the lac operon present. So here's lac Z, Y, and A, the 3 structural genes of the operon. And we can have this carried on the host chromosome, let's say, and on the plasmid DNA, we could have carried, could say this corresponds to this, we could have carried on the plasmid different alleles of the lac operon. So here, for example, we could have lac I wild type. Here's a promoter, and here's an operator. And then we could have, for example, a mutation, let's say in the Z gene. And we'll say we have a normal Y gene here. Let's say these are all wild type in the chromosomal copy. And here we have a mutation in the lac Z gene. Here we have a wild type lac Y gene and a wild type lac A gene. So we are, in this case, partially diploid, and we could say that, for example, for the Z gene, we are heterozygous for the lac Z gene. So in these diploids, then, we can create different states, different situations, different allelic combinations that represent the diploid state. And thereby we can combine wild type, and we can make heterozygotes, if you will, for wild type and mutant alleles. And this is what the technique that, one of the key techniques that Jacob-Monod used to ferret out the mechanism of the genetic switch that turns the lac operon on or off. And this really was the first time that any biologist had deciphered the mechanism of throwing a genetic switch, turning genes on and off. And Jacob-Monod won a Nobel Prize for their work. So let's go over some of their data now, because it is such an important experiment in the history of biology and is really an elegant approach using genetics to decipher a complex mechanism. So here we are looking at various genotypes in this left-hand column. And then we are assaying the gene products coming from the LAC-Z gene and the LAC-Y gene. That is, the LAC-Z gene encodes the enzyme beta-galactosidase, and the LAC-Y gene encodes the protein permease. And we can assay for the activity of beta-galactosidase and permease. There are biochemical assays that can determine whether those proteins are present. That is, whether they have been manufactured by the bacterial cell. And pluses indicate that, for example, beta-gal is active, and minus would indicate that beta-gal is inactive. And the same holds for the permease enzyme, another product of the lac operon. And IPTG is a non-hydrolyzable analog of the sugar lactose. So when we have IPTG present, just think of that as lactose. But the lactose, in this case, the lactose concentration will not decrease as time goes on, because IPTG is non-hydrolyzable. So we can have IPTG present or IPTG absent, present or absent. And the IPTG then acts like lactose as an inducer of the operon by causing the repressor to lose affinity for operator DNA. So they figured all this out by making different genotypes and observing the phenotypes that are produced. So we're going to go through these four first, and then we'll go into some marrow-diploid genotypes and see what the phenotypes are there. So let's go through this process now. Here we have in our first row a completely wild-type chromosome. We have a wild-type repressor gene. We have wild-type LAC-Z gene that encodes the enzyme beta-galactosidase. And we have a wild-type LAC-Y gene which encodes the enzyme or the protein permease, which brings lactose into the cell. And as might be expected, when the inducer, that is lactose or the lactose analog, is present, the operon is turned on because the repressor has lost affinity, specific affinity for operator DNA. And so both permease is made and beta-gal is made when the inducer is present, the lactose analog is present, but they are not made, those products are not made when the inducer is absent, which makes sense if you understand the model thus far. And we call that phenotype the LAC wild type, the LAC plus phenotype. That's the normal phenotype. Now, Jacob-Monod obtained a mutation which caused a constitutive LAC phenotype. That's the LAC-C phenotype. And all constitutive means is that it's on all the time, as you can see by the presence of beta-gal and permease, two products of the lac operon, even when inducer is not present, even when the lactose analog is not present. So the operon is turned on always here, and that we have a lac constitutive phenotype. And the mutation that they dealt with here mapped to the LAC-I gene. That is, it mapped to the repressor gene that encodes the LAC repressor. Now here is a mutation which is the LAC minus phenotype, which produces the LAC minus phenotype. And this mutation also maps to the LAC-I gene, the gene that encodes the repressor. But in this case, the LAC minus phenotype reflects an inability to manufacture operon products even when inducer is around. And we have to remember that Jacob-Monod did not have their model when they obtained this data. This is the data that led to their model. So we'll interpret these phenotypes in the context of the model shortly, but let's look at some more genotypes and phenotypes associated with them. Here is another genotype that yields a constitutive phenotype, just like the constitutive phenotype caused by a mutation in the second row genotype shown here. That is a constitutive mutation in the LAC-I gene. But this mutation didn't map to the LAC-I gene. It mapped to the operator region of the operon. It mapped very close to the structural genes of beta-gal and permease. So here we have another constitutive phenotype in which the operon is on all the time because gene products are observed even in the absence of inducer, even in the absence of the lactose analog. And we have a constitutive phenotype, but again, this is a mutation that is in a different part of the genome, and different part of the DNA. It maps to operator, and whereas this maps to the repressor gene. So let's look at some other genotypes here. And now I'm giving you marrow-diploid genotypes here. So here we're combining one strand of DNA on the chromosome or on the plasmid. It doesn't matter how you visualize this. So here we have one strand of DNA, which is wild-type LAC-I, mutant for both Z and Y. That is, these genes are incapable of producing functional permease Y or beta-gal Z. And that is over a chromosome or a plasmid. We have the other part of the diploid here is the LAC-I constitutive mutation that we considered in the second row up here, and wild-type genes for Z and Y. So these wild-type genes, Z and Y, are capable of producing functional beta-galactosidase and permease. So let's go through this, and we see that we have a LAC wild-type phenotype. That is, the operon is regulated appropriately. It's on in the presence of inducer and off in the absence of inducer, and that holds true for both of these gene products. So we can conclude in this situation that the LAC-I wild-type allele is dominant to the LAC-I constitutive allele. And not only is it's dominant in this case, it's dominant not only on gene affecting genes on the same strand of DNA, it's dominant in that it can appropriately regulate these wild-type genes found on another strand of DNA, on plasmid DNA, let's say, because these mutant alleles of Z and Y are incapable of producing any functional gene product, but these are the wild-type alleles. So in this case, it appears that the LAC-I gene product is capable of regulating the operon on another strand of DNA. That is, it seems as though the LAC-I wild-type allele produces a diffusible gene product. The LAC-I allele produces a diffusible gene product that can diffuse to another strand of DNA and regulate those genes appropriately in a wild-type fashion. And that it not only can do that, but of course it can also regulate, we might expect that if it can do that, it can regulate wild-type genes found on the same strand of DNA that the wild-type allele is at. So the LAC-I wild-type allele is dominant to the LAC-I constitutive mutation in both cis and trans. And by cis we mean the same strand of DNA, it's acting in cis, whereas in trans it's acting on a different strand of DNA, genes on a different strand of DNA. And therefore, Jacob and Monod could conclude that the LAC-I gene is actually producing a diffusible product. Now we'll look at the model and see how that data fits even better. But first let's go again through some other genotypes. The conclusion so far, though, is that the LAC-I gene, whatever it was, encodes a diffusible substance that can appropriately regulate genes on the same strand of DNA, shown here, or on a different strand of DNA, shown here. Well, now let's look at another marrow-diploid genotype. In this case, we are heterozygous for the LAC-I wild-type gene, allele, and a LAC-I S allele, which, as you remember, in the haploid state produced a LAC- phenotype where the operon was not turned on even in the presence of inducer. And apparently the same thing is happening here. We have a LAC- phenotype, meaning that the LAC-I S allele is dominant to the LAC-I wild-type allele. Now, if the lactose IS allele is producing a diffusible substance like the wild-type allele of LAC-I+, as we saw up in the previous two rows, then something about that diffusible product is unable to allow the activation of the operon. And keep that in mind now when we return to the model that we're going to go over. So let's look at another genotype here, another couple genotypes. Here we now are focusing in on the operator mutations, specifically the lac operator constitutive mutation that produced a situation in which the operon was always on, whether inducer was present or absent. The operon was always on, producing gene products. And here we've combined the constitutive operator mutation with the wild type operator. But we've done so in a way that allows us to determine whether or not the operator is also producing a diffusible substance that regulates the lac operon, as we saw for the LAC-I gene. In this case, what we see is that the LAC-OC, the constitutive mutation, the constitutive allele of operator, is dominant in cis, but is not dominant in trans. So let's look at that now. Here we see we have wild-type gene products. We have wild-type genes, wild-type alleles for the Z and Y genes, which produce, again, respectively, beta-galactosidase and permease, are assayable products of the operon. And on the other strand of DNA, we have wild-type allele of O, and we are mutant for Z and Y. That is, these alleles produce no functional beta-galactosidase or permease. And what we see here is a lac constitutive phenotype. That is, the constitutive allele of the operator gene, if you want to call it that, it confers an inability to turn the operon off even when inducer is absent. So the operon is on even when inducer is absent. Now we're combining a little bit of a different situation here. By making heterozygotes for Z plus and Z minus and Y plus and Y minus on different strands of DNA, we can determine whether this constitutive mutation of that maps to the operator acts only in cis or can act in trans as well. And what we find is that in this case, the Z gene, the Z gene of the operon is always on. It's constitutively on. But the Y allele here, the wild type allele of Y, is regulated appropriately. So here what we see is that the LAC-O wild type allele is regulating the Y gene appropriately, because it is on the same strand of DNA as the wild type allele of Y. Whereas here, the constitutive mutation of the operator is unable to regulate genes on the same strand of DNA appropriately. So C is dominant in cis, but cannot act in trans, does not act in trans to cause misexpression of the operon on it when the genes that are being regulated are a different strand of DNA. So here we have a contrasting situation. We have clearly a LAC-I gene, which is producing something diffusible that can regulate the operon appropriately or inappropriately, depending on the mutant or wild-type allele that we're talking about. Whereas operator mutations can only act in cis. They only act on the genes that are on the same strand of DNA as the particular allele that it is we're looking at. Here, wild type regulates Y appropriately, whereas C turns on Z plus constitutively, shown here, and Y being regulated appropriately, shown here. Finally, we see that the LAC-O constitutive allele is dominant in cis, even when the LAC-I S allele is present. So now we're looking at a more complicated situation. Here on one strand of DNA, we have wild-type LAC-I gene, wild-type LAC-O gene, and wild-type Z and Y genes. On the other strand of DNA, we have the LAC-I S allele, which, as you'll remember, is dominant in marrow-diploids to the LAC-I wild-type allele, as shown by this LAC minus phenotype. Here we have LAC-IS on this strand of DNA with LAC-OC constitutive mutation, in other words, in the O in the operator, and wild-type Z plus and Y plus genes. And the phenotype is a LAC constitutive phenotype, meaning that the operon is always on. So even in the presence of LAC-IS, which turn the operator off under all circumstances, we have the operator on in all circumstances if we have a mutant operator, a constitutive mutant in the LAC-O gene. So what these elegant experiments of Jacob and Monod showed is that there are two key regulatory elements that determine whether the operon is on or off. That is, two key elements that throw this genetic switch one way or another. There is the LAC-I element, which maps at a short distance away from the operon but is not adjacent to the structural genes of the operon. And then there is the operator element, which they call O, the operator element, which maps very close to the structural genes of the operon. And both of these elements are very important to the regulation of the operon, but one, that is LAC-I, produces a gene product which is diffusible and which regulates the operon even in trans, that is, can regulate operons on other pieces of DNA, other strands of DNA, whereas the operator is an element, a regulatory element that acts only on the same strand of DNA in which it is found. So we'll come back and look at these interpretations of these phenotypes, but let's first just talk about the model a little bit and explain how they interpreted their data again. So we'll see how they derived their model right here. So let's imagine that there were mutations in the operator DNA that changed the nucleotide sequence of the operator. What would happen to the regulation of the operon? Well, if operator nucleotide sequences were changed, then the repressor would not be able to bind the operator with high affinity anymore, because the repressor contains a DNA binding domain with amino acids that recognize the nucleotide sequence present in operator DNA. So if operator mutations were present, you would expect the operator to be what? On or off. So if you had the operator constitutive mutation adjacent to, let's say, a wild type Z and a wild type Y gene, the phenotype that you would expect is that the operator would be on, always on, or constitutive. And it would be always on by virtue of the fact that the repressor can't bind the operator anymore, whether or not lactose is present or not. So the operon is on even when lactose is absent. When the operon should be off, the operon is still on because repressor can no longer bind the operator because of the mutation, the constitutive mutation in the operator. Well, what about the LAC-I mutants that we looked at? If we have a LAC-I constitutive mutation, that produces a diffusible product which can diffuse and cause the operon to be on at all times, whether or not lactose is present or not. So what do we imagine there has happened to the product of the LAC-I gene? Since it produces a diffusible product, probably a protein, the LAC-I constitutive allele represents a repressor which can't bind operator DNA because the LAC-I constitutive mutation produces a phenotype where the operon is always on. It's constitutive, in other words, and that's by virtue of the repressor protein can't bind the operator. Not because there's been a mutation in the operator in this case, but because the protein product of the LAC-I gene has been altered in its DNA binding domain such that it can no longer recognize in a specific manner operator DNA. And therefore, the operon will always be on. will always be turned on. And that's the basis of the LAC-I constitutive phenotype. And we can see that there are other types of mutations of the LAC-I gene, though. Remember that we have LAC-IS. And LAC-IS, you will remember, resulted in a LAC- phenotype. So LAC-IS produces a LAC- phenotype. That is, no operon expression occurs, even if lactose is present. Even if lactose is present. So in the normal case, when lactose is present, that causes the repressor to lose its specific affinity for operator DNA, although it has some DNA binding affinity, but not specifically for operator. It doesn't bind the operator with any high affinity when lactose is present, essentially causing the repressor to fall off, we'll say in loose terminology, to fall off the operator or to slide along the DNA non-specifically. But what if the LAC-IS mutation produced a defective allosteric binding site for lactose so that the repressor here, the repressor can't bind lactose? If the repressor protein cannot bind lactose, then it will never achieve its low affinity state for operator and will remain bound tightly in a specific manner to operator DNA, thereby shutting off the operon under any circumstance, whether lactose is present or not. So by an elegant series of genetic experiments that identified two regulatory elements, the operator and the repressor gene, operator simply being interpreted as a DNA sequence, which does not encode any gene product, but rather is a binding site for the product of the LAC-I gene, a repressor protein, which is responsible for turning the operator off unless the inducer of the operon, lactose, is present. These two regulatory elements work in concert then to throw this genetic switch on or off. And the mechanism for that was determined by Jacob and Monod using a series of elegant experiments that showed that LAC-I produces a diffusible repressor protein and operator DNA, which serves as a binding site for that diffusible repressor protein. And then it becomes possible to understand these series of genotypes and their phenotypes. So what I'm going to suggest to you is that you go through some of these and employ your understanding of the model and work backwards to understand how these different genotypes produce the various LAC phenotypes that we've considered. And when you do that, you will see that the model of Jacob and Monod makes perfect sense and accords very nicely with this data. And naturally, this genetic data was combined with firm biochemical data that followed, which showed indeed that this is the mechanism for lactose operon regulation, the primary mechanism for lactose operon regulation. And Jacob and Monod deservedly won a Nobel Prize for this work because this was the first time that any geneticist, any biologist had understood how it is that genes are turned on or off in any organism. We could take as our example here this last genotype just to coordinate our understanding of the model with the data obtained in this genotype. So let's look at that very briefly. Here we have the LAC constitutive phenotype. It's always on. And we can say, well, I know why it's always on, even when there's a mutation in the repressor gene, the LAC-I gene, which produces a repressor that cannot bind lactose and therefore should remain permanently bound to operator DNA. Well, that's probably true. It probably shuts down this strand of DNA here, this top operator strand of DNA, because it probably is permanently tightly bound to the wild type operator. But even though the IS repressor gene product is incapable of binding wild type, or is always going to be bound to wild-type operator DNA. It's incapable of binding to the mutant constitutive operator because the nucleotide sequence of the operator has been altered in such a way that prevents the repressor DNA binding domain from recognizing it and binding it. And therefore, these wild-type gene products here are being produced by the Z plus and Y plus gene on this bottom strand of DNA that we're showing here in this marrow diploid. So that's just an example of how we can reverse engineer, if you will, Jacobi Monod's thinking on this matter. And now what we're going to move on to in the next part of this lecture is a little more complicated, or some complications to regulation of the lactose operon, and look at the regulation of lactose operon in the presence of a superior food source as compared to the lactose sugar. And that's where we'll pick up with in the next part of this lecture.